All right, thank you. So you might be asking why someone in agriculture from the Agricultural Research Service will be talking about strokes, and I'd say that's a really good question. There's no answer. But I do work for the Soil, Plant, and Water Research Center in Florence, South Carolina, and so that's actually where our tie-in comes. But I actually started as a medical technologist, and so when you work in the health professions, you find that when you mention that, people start talking to you about their conditions and things like that, and so you get a little bit more about people than you probably really want. So when I first started working, I came from Illinois, and when I first started working in South Carolina, my research leader came to me and he said, "Congratulations, you now work in the stroke belt." And I said that was not in the recruitment brochure. And we got to talking about that, and so the stroke belt, as that map shows, runs basically from Virginia down through into Louisiana, and Florida has its own little set of crazy, so it was actually spared. But more than that, South Carolina actually resides in what is known as the buckle of the stroke belt, and so people there have not only higher incidences of stroke than the rest of the United States, but it also has higher incidences of stroke even for those that live in the stroke belt itself. And it's not just the incidences, it's actually even early onset. So it's not uncommon to read the news and hear about somebody who's in their mid to late 30s having strokes. And so much so that actually, if you want to specialize in strokes, residents from all over the country will come to South Carolina and study in this area. And so there's a lot of questions on why we have these increases of stroke rate in this area, and we don't know. People have looked at a number of hypotheses, and all of them have been proven to be unproven. And it is not, as we would believe, yes, there are cases of hypertension, and there are things that you can do with your diet, but those do not explain the increased incidences. So it is not socioeconomic, and it is not genetic, and we don't know. But what we do know is actually that if you look at the excess stroke risk, it is for people who lived in the stroke belt during childhood that implicates it implicates early life exposure. And so what we know is that if you are, like my two boys now that have been born in this area, if you spend the first couple years of your life in the stroke belt, and then you move outside of that area, so say you move to the other end of the country, you carry this increase in stroke risk with you for the rest of your life. Now, someone who was born in New York and moves into this area, my stroke risk, I could spend the next 60 years of my life, and my risk will never approximate what my boys would, even just living there for 2 years. So we know it's carried with them for the rest of their life. And so how does such an early life exposure event occur? Now, I think that Dr. Nachman's slide talking about how soil moves around kind of shows us that soil can move through systems. And I think at this point now we all know that children love to ingest things. And so it's really not very far-fetched to understand that ingestion and inhalation are both very easily acceptable methods for this exposure event, and that it can easily be tied to the local environment as well. When we also understand that the human gut takes approximately a year to develop after birth, we can understand that there is potentially some sort of introduction that can establish early on in the life of a child that they can then carry with them for the rest of their life. Now, I'm a microbiologist, so I hit everything with the bacteriological hammer, so I will everything from this point forward is from a microbiological perspective, but that doesn't mean that there can be some other instance. It could be a chemical, it could be viral, for example. So in these talks with my boss and just talking about stroke, it could have just ended there. But in just looking at things, it turns out that South Carolina and the United States, the southeastern United States, isn't the only place that has a stroke belt. And so the middle of this slide here actually looks at England, which also does have a stroke belt as well. And so if you live in southern England, you have your lower incidences of stroke mortality. And then from Wales up into the southern part of England, you have a higher risk. And then for those who are, unfortunately, the Scottish suffer from the highest rates of stroke mortality. So I thought it was pretty interesting, and we kind of left it at there until about 2011. And then an interesting paper that looked at the biogeography of England was published. And so that's on the left-hand side. And if you actually shade that instead of orange, you use blue, you notice that there is a pretty striking similarity between the two. And so I saw that, and I went to my boss, and I said, remember we were talking about strokes? I think we have something here. And so actually, if you look at that, they did a study, and throughout the study, and they looked at what drivers were there in bacterial populations. And so obviously, really doesn't come as much a surprise, but it's mostly moisture and pH. And those, I mean, anybody that studies microorganisms knows that if you really want to adjust microbial populations, adjust one of those two, and you'll see a significant change. And so the map on the far right is the pH scale of England, and so you can see a good correlation between all three of those right there. And so we said, hey, let's look at what we see in South Carolina. And so we actually went to one of the preeminent researchers, Daniel Lackland, who does the same thing with strokes in South Carolina. And so we had the mortality data. And we can see basically right across I-95, we get from the east, we see increases in strokes. And then when we go into the upland of South Carolina, there are a couple of counties that have higher incidences, but predominantly it all resides on the East Coast. And that corresponds with the coastal plain areas. And so then we looked at the soil data, and we noticed that there was a very strong correlation between depth of water table and a lot of the hydric ratings of soil. And all this data is from NRCS. And so we see a good correlation with depth of water table. So the higher the water table is to the surface, anaerobic conditions, for example, we see a good correlation. And then pH as well is very strongly. The more pH, the more acidic your soil gets, the better it correlated with the stroke data. Unfortunately, we have no biological data. There is no biogeography of the South Carolinian soils. And so our hypothesis, though, is there's a correlation between stroke rate and environmental microbes. And so we have all the existing data points to show for an initial investigation. So whether it was chemical or microbiological, now we know the perfect areas to go and look at it. But we're lacking that biogeochemical sketch and the microbial fingerprint. So how do you get that? Well, there's a couple of ways. The first, now TRFLP is highlighted because that was the method that the British used, but it tends to be nonspecific. And so actually in 2018, the French did a similar study. They looked at the biogeography of France, and they did DNA sequencing. Now, that does increase the costs a bit, but the fact that nowadays the sequencing is generally inexpensive, I think that is really a potential route that we could go. Now, the question is it existence of a pathogen? And people have said, well, maybe it's the absence of a protectorant. And so now we go from whether it's a needle in a haystack or whether it's a missing needle in several haystacks. So it gets to be a little bit problematic. What are the intended results and impact of this research? Well, best case scenario, we reduce the threat that stroke plays in the Southeast, we win a Nobel Prize, and everybody's happy. Worst case scenario, though, is that we have better understanding of South Carolina biogeography. I think it's interesting that I can tell you more about the microbial content of Antarctic soils than I can of non-ag soils in South Carolina. I mean, there is no data in non-ag soils of South Carolina. And actually, there's very little microbial data on any non-ag soils throughout the whole United States. It's missing, and we need to address that. Now, for the biogeography research team, obviously, statisticians, epidemiologists, microbiologists, but I want to mention NRCS. NRCS runs the gamut of all the soils data that we have in the United States. They do a great job. It's all physical and all chemical, but there's not a lot of biological data. Actually, there's almost no biological data. Is anybody from NRCS here? Okay. Well, I'm I'm nominating you guys to be the database for all of the future microbiome state. National Resource Conservation Service. Yeah. So, but additionally, if we're going to do this, we need to get the South Carolina Department of Natural Resources and other state and federal agencies to get land access. Now, I'm the type of person that when I walk into a lab, I say this experiment's going to be easy to the chagrin of everybody in my lab. But really, I think we could do the sequencing of South Carolina in about 6 months. It's what they did in the United Kingdom, and South Carolina is about a third of the size. And then an additional 6 months of lab analysis. Never mind the year or so that it would take to plan to do all this. Resources, I'm not going to mention money. We just need a lot of manpower. And then funding. Really, one of these projects really is one of those instances where I think that, given the medical connotations, really something like NSF or NIH would need to be involved. So thank you for your time.